Good day everyone, my name is Jocelyn T. Manibo, your reporter today. Now I'm going to discuss the nature of the gene and the genome with a subtopic of the concept of a gene as a unit of inheritance, chromosomes the physical carriers of the gene, the chemical nature of the gene, the structure of the genome, the stability of the genome, sequencing genome the genetic basis of the human, with the human perspective application genomic analysis to medicine and experimental pathways the chemical nature of the gene. This figure is an overview depicting several of the most important early discoveries on the nature of the gene. Each of these discoveries is discussed in this present chapter. First, we are going to discuss the concept of a gene as a unit of inheritance. The science of genetics began in the 1860s with the work of Gregor Mendel, a friar at the Abbey of St. Thomas located in Thomas Czech Republic. Mendel too began his studies, but he eventually had a clear experimental plan in mind. His goal was to mate or cross pea plants having different inheritable characteristics and to determine the pattern by which these characteristics were transmitted to the offspring. Mendel chose the garden pea for a number of practical reasons, not the least of which was he could purchase a variety of seeds that would give rise to plants with distinct characteristics. Mendel chose to focus on seven clearly definable traits including plant's height and flower color, each of which occurred in two alternate and clearly identifiable forms. Mendel crossbred plants through several generations and counted the number of individuals having various characteristics. After several years of research, Mendel drew the following conclusion, which are expressed below in modern genetic terminology. The characteristics of the plants were governed by distinct factors or units of inheritance, which were later termed genes. An individual plant possessed two copies of gene that controlled the development of each trait, one derived from each parent. The two copies could be either identical to one another or non-identical. Alternate forms of a gene are called alleles. For each of seven traits studied, one of the two alleles was dominant over the other. When both were present together in the same plant, the existence of the recessive allele was masked by the dominant one. Second one is each productive cell or gamete produced by a plant contained only one copy of a gene of each trait. A particular gamete could have either the recessive or the dominant allele for a given trait, but not both. Each plant arose by the union of a male and female gamete. Consequently, one of the alleles that govern each trait in a plant was inherited from the female parent, and the other allele was inherited from the male parent. Third is, even though the pair of alleles that govern trait remain together throughout the life of an individual plant, they become separated or segregated from one another during the formation of the gametes. This finding formed the basis of Mendel's law of segregation. The fourth is the segregation of the pair of alleles for one trait had no effect on the segregation of alleles for another trait. A particular gamete, for example, could receive a paternal gene governing seed color and a maternal gene governing seed shape. This finding formed the basis of Mendel's law of independent assortment. Observation of dividing cells by the German biologist Walter Fleming in the early 80s revealed that the cytoplasmic contents were shuttled into one daughter cells or the other a matter of chance, depending on the plane through which the furrow happened to divide the cell. In contrast, the cell appeared to to go to treat lengths to divide the nuclear contents equally between the daughters. During cell division, the material of nucleus became organized into visible threads which were named chromosomes, meaning colored bodies. This figure show the events occurring the roundworm ascaris following fertilization. As reported in a classic 19th century investigation, both the male and female gamete are seen to contain two chromosomes. Fusion of the sperm and egg nuclei called pronuclei in the egg cytoplasm between E and F produces a zygote containing four chromosomes. The second polar body shown in A is a product of the previous meiosis. The importance of the chromosomes contributed by the male became evident in a study by the German biologist Theodor Boveri on a sea urchin eggs that had been fertilized by two sperm rather than just one as is normally the case. This condition, referred to as polypermy, in characterized the destructive cell divisions and the early death of the embryo. The second sperm donates an extra set of chromosomes and extra centriole. These addition components lead to abnormal cells division in the embryo during with the daughter cells receive variable numbers of chromosomes. Vaubery concluded that orderly process of normal development is dependent upon a particular combination of chromosomes, and this can only mean the individual chromosomes must possess different qualities. This was the first evidence of qualitative difference among chromosomes. In 1883, it was noted by the Belgian biologist Edward van Beneden that the cell of the worm's body had four large chromosomes, but the male and female nuclei presents in the egg just after fertilizations, or before the two nuclei fuse with one another, had but two chromosomes apiece. About the same time, the process of meiosis was described, and in 1887, the German biologist August Wiesmann proposed that meiosis included a reduction division, during which the chromosomes number was reduced by half prior to formation of the gametes. If a reduction division did not occur and each gamete contained the same number of chromosomes as in adult cell, then the union of two gametes would double the number of chromosomes in the cells of the progeny. The number of chromosomes would double with each and every generation, which obviously does not and cannot occur. This figure is the homologous chromosomes that the Walter Sutton, a student at Columbia University, published a paper that pointed directly to the chromosomes as the physical carriers of Mendel's genetic factors. Sutton followed the development of sperm cells in the grasshopper, which, like Ascaris, has large, easily observable chromosomes. Sutton's drawing of the homologous chromosomes of the male grasshopper that have associated during the meiotic prophase to form bivalent 11 pairs of homologous chromosomes, A to K, and an unpaired X chromosomes were observed. The chromosome as a linkage group that we are all know that have a different linkage group that is different chromosomes. As it happens, the garden pea has 7 pairs of chromosomes. The genes that govern each of Mendel's traits either occur in different chromosomes or are so far apart from one another on the same chromosome that they act independently. Sutton's prophecy of linkage group was soon confirmed. Within a couple of years, genes for two traits in sweet peas, flower color and pollen shape, were shown to be linked. Other evidence for chromosomal linkage rapidly accumulated. Take note that genes on the same chromosome should act if they are linked to another. That is, they should be part of the same linkage group. This figure is a Drosophila, or the fruit fly, that have a generation time from egg to a sexually mature adult of about 10 days and can produce up to 1,000 eggs in a lifetime. That the fruit flies are a very small, so large number could be kept on hand. They were easy to house and breed, and they were very inexpensive to maintain. In 1909, fruit flies seemed like the ideal organism to Thomas Hunt Morgan of Columbia University, and he began that was to be a start of a new era in genetic research. In beginning work with the insect, there was only one strain of fly available, the wild type. Whereas Mendel had simply purchased various of pea seeds, Morgan had to generate his own variety of fruit flies. Morgan expanded that variants from the wild type would appear in he bred sufficient numbers of flies. Within a year, after observing thousands of flies, he found his first mutant, that is, an individual having an heritable characteristic that distinguished it from the wild type. The mutant who had white eyes rather than the normal red color ones. By 1915, Morgan and his studies had found 85 different mutants with a wide variety of affected structures. It was appeared that on rare occasions a spontaneous change or mutation occurs within a gene alternating independently, so that the altered trait is based from generation to generation. As you can see, this figure is the fruit fly Disophilia melangaster. This photograph of a wild female fruit fly and a mutant male that carries a mutation leading to white eyes. This figure is called the crossing over and recombination. In 1911, Morgan offered an explanation for the breakdown in linkage. In 2 years earlier, F.A. Janssens had observed that the homologous chromosomes of behaviolants became wrapped around each other during meiosis. Janssens had proposed that his interaction between maternal and paternal chromosomes resulted in the breakdown and exchange of pieces. Capitalizing on the proposal, Morgan suggested that this phenomenon, which he termed crossing over, or the genetic recombination, could account of the appearance of offspring or the recombinants having unexpected combination of genetic traits. An example is this figure that the fruit flies have 4 pairs of homologous chromosomes, one of which is very small. The two dissimilar homologous are the chromosomes that determine sex, as in human. Male fruit fly are XY and the females are XX. In analysis of offspring from a large number of crosses between adults carrying a variety of alleles on the same chromosome, indicated that the percentage of recombination between a given pair of genes on a chromosome, such as eye color and wing length, was essentially constant from one experiment to another, and the percentage of recombination between different pairs of genes, such as between eye color and wing length versus eye color and body color, could be a different. In this figure showing the crossing over provides the mechanism for reshuffling alleles between maternal and paternal chromosomes. A is the bivalent or the tetrad formation during the meiosis, showing three crossing intersections or the chiasmata indicated by red arrows. B is simplified, the representation of a single crossover in a Drosophila heterozygotes. B and W at chromosomes number 2 and the resulting gametes if either of the crossing gametes participants in fertilization, the offspring will have a chromosome with a combination of alleles that was not present as single chromosomes in cells of either parent. In 1911, Alfred Sturtevant, an undergraduate at Columbia University who was working in Morgan's laboratory, conceived the idea that the recombination frequencies could be used to map the relative positions of individual genes along a given chromosomes. An example of the underlying principle of this mapping procedure is illustrated in this figure. That in this example, the genes for wing length and the body color in Drosophila are situated at considerable distance from one another on the chromosome and therefore are likely to become unlinked by an intervening break, breakage and the crossover. In contrast, the genes for eye color and body color are situated very close to one another on the chromosome and consequently are less likely to become unlinked. Using recombination frequencies, Sturtevant, who became one of the century's most prominent genetics, began to construct detailed maps of the serial order of genes along each of these four chromosomes of the fruit fly. The recombination frequencies have since been used to prepare chromosomal maps from viruses and bacteria, as well as a large variety of eukaryotic species. This figure showed the mutagenesis and the giant chromosomes, that giant polyethylene chromosomes of larval insects. This Figure A, these giant polyethylene chromosomes from the salivary gland of a larval fruit fly show several thousand distinct darkly staining bands. The bands have been identified as the loci of the particular genes. The insect shows how polyethylene chromosomes consist of a number of individual DNA strands. The stained bands on the chromosomes correspond to sites where the DNA is more tightly compacted. The second figure is the scanning electron micrograph of a giant polyethylene chromosomes from a Crenonomus larva, showing how specific sites are expanded to fruit aphid chromosomes are sites where DNA is being very actively transcribed. Unusual polyethylene chromosomes, as they are called, are rich in visual detail, displaying about 5,000 bands. When stained and examined microscopically, the banding pattern is essentially constant from one individual to the next, but differences are observed between the chromosomes from the flies of different species of the Drosophila genus. Patterson found the individual bands correlated with specific genes. The relative positions of these genes on the giant chromosomes agreed with those predicted on the giant chromosomes agreed that the basis of genetic maps prepared from the recombination frequencies that providing visual confirmation of the validity of the intermapping procedure. Next, we discuss the chemical nature of the genes. And The structure of the DNA was investigated by a number of laboratories in both United States and England in early 90s and was sold by the James Watson and Francis Crick at Cambridge University in 1953. Before describing their proposed DNA structure, let us consider the facts available at the time. Based on the composition, the basic building block of DNA was known to be a nucleotide consisting of 5 carbon sugar deoxyribose, to which one phosphate is esterified at the 5 position of the sugar ring and one nitrogenous base in attached at the one site. There are two types of nitrogenous base present in a nucleic acid. Uh, the pyrimidines, which contain a single ring, and purines, which contain two rings. The DNA contains two different pyrimidines, thymine and the cytosine, and the two different purines, guanine and the adenine. The nucleotides were known to be a covalent linked to one another to form a linear polymer or strand with a backbone composed of alternating sugar and phosphate groups joined by 3 to 5 phosphodiester bands. The bases attached to each sugar were thought to protect from the backbone like a column of struck shelves. T. M. Morgan proposed the gene theory, which states that chromosomes are bearers of hereditary units and each chromosome carries hundreds or thousands of genes. The second is the genes are arranged on the chromosomes in the linear order and on the special regions or the locus. This figure is the chemical structure of DNA. A is the model of the DNA nucleotide containing the base thymine, and the molecule is deoxythymine, and with the 5 monophosphate or DTMP. The net-like cage represents the electron density of the atoms that make up the molecule. B is the chemical structure of a DNA nucleotide containing the base adenosine, and the molecule is deoxydenosine 5 monophosphate or DAMP. A nucleotide is composed of a nucleoside linked to a phosphate. The nucleoside portion of the molecule is enclosed by the dashed line. C is the chemical structure of a small segment of single DNA strand showing all 4 nucleotides. Chargaff discovered the following rules of DNA-based decomposition. A is equal to T, G is equal to C, A plus T is or either G plus C. Chargaff findings put the DNA molecule in a new light, giving in specifically and individually from one organism to another. The significance of these base equivalences, however, remained obscure. The Watson-Crick proposal is the importance of secondary and tertiary structure as determinants of the protein's activity was stressed. Similarly, information about the three-dimensional organization of DNA was needed if its biological activity was to be understood using X-ray diffraction data. That Watson-Crick proposed a structure of DNA that included the following elements. Number 1 is the molecule is composed of 2 chains of nucleotides. That this conclusion followed on the heels of an erroneous proposal by Linus Pauling, who had suggested that DNA was composed of three nucleotide strands. The second one is the two chains spiral around each other to form a pair of right-handed helices. That in a right-handed helix, an observer looking down the central axis of the molecule would see that each strand follows a clockwise path using away from the observer. The helical nature of DNA was revealed in the pattern of spots produced by Franklin X-rays diffraction image, which was shown to Watson during a visit to King's College. In number 3 is the two chains comprising one double helix run in the opposite direction, that is, they are antiparallel. Thus, if one chain is aligned in 5 to 3 direction, its partner must be aligned in 3 to 5 direction. Number 4 is the sugar-phosphate backbone of each strand is located on the outside of the molecule with the two sets of bases projecting toward the center, that the phosphate groups give the molecule a large negative charge. And number 5 is the bases occupy planes that are approximately perpendicular to the long axis of the molecule and are plates, or therefore stack one off top of another, link a pile of plates. The hydrophilic interaction and van der Waals forces that between the stacked planar bases provide stability for the entire DNA molecule, the molecule to resemble a spiral staircase. This matter of construction is evident in the chapter opening photograph, which shows the original Watson-Crick model. And number 6 is the two strands are held together by the hydrogen bonds between each base on the strand and the associated base on the other strand. Because of the individual hydrogen bonds are weak and easily broken, the DNA strands can become separated during various activities, but the strand of hydrogen bonds are additive, and large numbers of hydrogen bonds holding the strands together make the double helix a stable structure. Number 7 is the distance from the phosphorus atom of the backbone of the center of the axis is 1 nanometer. Thus, the width of the double helix is 2 nanometer. It as the pyrimidine in one chain is always paired with a purine in the other chain. This arrangement produces a molecule that is 2 nanometer wide along its entire length. 9 is the nitrogen atoms linked to carbon 4 of cytosine and carbon 6 of adenine are predominantly in the amino or NH2 configurations, that rather than the amino or NH form. Similarly, the oxygen atoms linked to carbon 6 of guanine and carbon 4 of thymine are predominantly in a keto or the configuration rather than the enol form. These structural restrictions of the configuration of the bases suggested that adenine was the only purine structurally capable of binding to thymine, and that guanine was the only purine capable of binding to cytosine. Therefore, the only possible pairs were A is to T and G is to C, which fits perfectly with the previous base composition analysis by Chargaff. A is to T pairs were joined by two hydrogen bonds. G is to C pairs by three hydrogen bonds. Number 10 is the spaces between adjacent turns of the helix form two grooves of different width. A wider major grooves and the more narrow mirror groove, the spiral ground the other outer surface of the double helix. Proteins that bind to DNA often contains domains that fit into these grooves. In many cases, a protein bound in one groove is able to read the sequences of the nucleotides along the DNA without having a separate dot strands. Number 11 is the double helix makes one complete turn every 10 residues 3.4 nanometer or 1.50 turns per million daltons of molecular mass. Number 12 is because an A on one strand is always bonded to a T on the other strand and G is always bonded to a C. The nucleotide sequences of the two strands are always fixed relative to one another. This relationship between the two chains of the double helix is referred to as complementarity. For example, the complementary to T is the 5, AGC is complementary to 3TGC is to 5, and one entire chain is complementary to the another. As we shall see, complementary is of the overriding importance in nearly all the activities and the mechanism in which nucleic acids are involved. This two figure is a double helix or the continued. C figure is the Watson-Crick base pair, and the figure is the electron microphage of DNA being released from the head of a 2 T2 bacteriophage. This linear DNA molecule that note the two free ends that measured at 68 macrometer is length, approximately 60 times longer than the page head in which it is contained. The importance of the Watson-Crick proposal is from the time biologists first considered DNA as the genetic material. There were the three primary functions it was expected to fulfill. The number one is the storage of genetic information. As genetic material, DNA must contain a stored record of instruction that determine all the inheritable characteristics that an organism exhibits. In molecule terms, DNA must contain the information for the specific order of amino acids in all the proteins that are synthesized by an organism. Number two is the replication and inheritance. DNA must contain the information for synthesis of the new DNA strand or the replication. DNA replications allows genetic instructions to be transmitted from one cell to its daughter cells and from one individual to its offspring. Number 3 is the expressions of genetic storage. The DNA is more than a storage center, it is also a director of cellular activity. Consequently, the information encoded in DNA has to be expressed in some form that can take part in events that are taking place within the cell. More specifically, the information in DNA must be used to direct the order by which specific amino acids are incorporated into a polypeptide chain. The three function required of the genetic material in this figure, that A is the DNA must contain the information that encodes inheritable traits. B is the DNA must contain the information that directs in its own duplication. And C is the DNA must contain the information that directs the assembly of specific proteins. The DNA supercalling explained by Jerome Vinogrand in 1963 and his colleagues at the California Institute of Technology discovered that two close circular DNA molecules of identical molecular mass could exhibit very different rates of sedimentation during configuration. Further analysis uh, indicated that the DNA molecules sedimenting more rapidly had more compact shape because the molecule was twisted upon itself, much like a rubber hand in which the two ends are twisted in opposition directions or the tangled telephone cord after extended use. The DNA in the state is said to be supercoiled because supercoiled DNA is more compact than its relaxed counterpart. It occupies a similar volume and moves more rapidly in response to centrifugal force or an electric field. Supercooling is best understood if one pictures a length a double helix DNA lying free on flat surface. A mole coil in the condition has the standard a number of 10 base pairs per turn of the helix is said to be relaxed. The DNA would still be relaxed if both ends of the strands were simplified fused to form a circle. Considered, however, what would happen if the molecule were twisted before the ends were sealed. If the length of DNA were twisted in direction opposite to in which the duplex was, would the molecule would tend to unwind. An underwound DNA molecule has greater number of base pairs per turn of the helix. Because the molecule is most stable with 10 residues per turn, it tends to resist the strain of becoming underwood by becoming twisted upon itself into a supercoiled configuration. DNA is referred to as negatively supercoiled when it is underwood and positively supercoiled when it is overwound. Circular DNA is found in nature or the mitochondrial, viral, or the bacteria are invariably negatively supercoiled. Supercoiling is not restricted to small or cellular DNAs but also occurs in linear eukaryotic DNA. For example, negative supercoiling plays a key role in allowing the DNA of the chromosomes to be compacted so as to fit inside the confines of microscopic cells nucleus. Because negatively supercoiled DNA is underwood, it exerts a force that helps separate the two strands of helix, which is required during both replications or the DNA synthesis and transcription RNA synthesis. The certain enzymes in both prokaryotic and eukaryotic cells are able to change the supercoiled state of a DNA duplex. The enzymes called typosomerases because they change the topology of the DNA. Cells contain a variety of typosomerases which can be divided into two classes. Type 1 topoisomerases change the supercoiled state of DNA molecule by creating transient break it once of the duplex. And the type 2 topoisomerases makes a transient break it both strands of the DNA complex, another segment of DNA molecule, or the separate molecule entirely, and then transported through the break and the severed strands are rescaled. This figure is the DNA topoisomerase. A is a model depicting the action of human topoisomerase 1. The enzyme has cut one of the strands of the DNA which is rotating around a phosphodiester band in the track strand. The cut strand will then be released. Note that the drawing depicts type 1b topoisomerase type 1a enzymes found in bacteria act by a different mechanism. B is a molecular model based on X-ray crystallography depicting the action of topoisomerase 2. In step 1, the dimeric enzyme is in an open conformation ready to bind the DNA segment. so named because it will form the gate through which the T to DNA and the segment will pass. In step 2, the enzyme has undergone a conformational change as it binds. The G enzyme has undergone of a conformation change as it binds the G segment. In step 3 and 4, the enzyme binds a molecule of ATP. The G segment is cleaved and the T segment is passed through the open gate. The back the backend the backted stage represents a hypothetical intermediate carrying out of the step in which the T segment is transported through the G segment. As this stage, both cut ends of the G segment are covalently bound to the enzyme. In step 5, the two ends of the G segment are rejoined and the T segment is released. ATP hydrolysis and release of ADTP and P are proposed to occur as the starting state is regenerated. And a C is the type of reaction that can be catalyzed by topoisomerasis. Part 1 shows supercoiling relaxation reactions. Part 2 shows unknotting reaction. Part 3 is shows ceteination decatenation reactions. The figure is the electron micrograph of a pair of interconnected that the cellular DNA molecules. Molecules of this type recollect uh, in bacteria lacking a specific typosomerasis. Next, we discuss the structure of the genome. Genome is the all of the DNA in a cell. All the DNA on all chromosomes includes genes and their genic sequence repeats. Especially, it is all the DNA in an organelle. Eukaryotes can have 2 to 3 genomes, and the nuclear genome is the mitochondrial genome, plasmid genome. If not a special genome, usually refers to the nuclear genome. And the genomics is the study of genomes, including large chromosomal segments that containing many genes. The initial phase of genomics aims to map and sequence an initial set of entire genomes. Functional genomics aims to deduce information about the function of DNA sequences. It should contain long after the initial genome sequences have been completed. But also, the human genome have 22 autosome pairs plus 2 sex chromosomes, 3 billion base pairs in haploid genome, about 3% codes for proteins, about 40 to 50% is repetitive made by the retro or transpositions. And what is the function of the remaining 50%? That were and what are the 30,000 to 40,000 genes that searching DNA for an open reading frame seems to be most logical way of finding genes, but just because an open reading frame existence does not definitely answer whether it is transcribed. In genomic resolutions, we also know that the close to all genes is or in the genome and that sequence of the proteins they encode. No longer look at just individual genes that examine whole genomes or systems of genes. This figure shows the absorption spectrum of thymine, all for the nucleotides of DNA and consequently DNA itself absorb maximally near 260 nanometer. The absorb stand at 260 nanometer is generally used to determine the nucleic acid concentrations. The ratio of absorb stands at 260 over 280 nanometer is one measured of the purity of nucleic acid preparations because contaminating proteins generally absorb most strongly at 280 nanometer. The 260 over 280 ratio of purified DNA is about 2. The second figure is the thermal denaturation of DNA. A thermal denaturation curve for native bacteriophage (TS) DNA in 0.3 million as sodium citrate. The melting of the DNA or the strand separation occurs over a narrow range of temperature, particularly for the simpler DNAs of small viruses. The temperature corresponding to half the increase in absorbed stance in term the TS form or from. This figure is the relationship between melting temperature and GC content. That electron micrograph showing a segment of sea urchin's DNA containing the genes of the 5 types of histone molecules. That those regions of DNA that undergo strand separations under the conditions employed are seen as thinner single strand segments. These regions corresponds to segments that are rich in 80 base pairs that lie between the DNA segments that code for the histone proteins. DNA renaturations, the separation of the two strands of the DNA duplex by heat, is not an unexpected finding, but the resociations of the single strands into stable double-stranded molecules with correct base pair seems almost inconceivable. However, in 1960, Julius Marmor and coworkers at Harvard University found that if they slowly cooled a solution of bacterial DNA that have been thermally denatured, the DNA regained the proteins of the double helix. It absorbed less ultraviolet light, once again behave like genetic material in being able to transform bacterial cells. And similar, the results were obtained by heating the DNA to 100 degrees Celsius to denature it, rapidly dropping the temperature of the solution to approximately 25 degrees Celsius below the T and the temperature, and allowing the DNA to incubate at this temperature for a period of time. It became apparent from this study that a complementary single-stranded DNA molecules were capable of associating an event termed renaturation or renilling. This figure is the kinetics renaturations of viral and bacteria DNAs. The curve shows the renaturations of shared strands of DNA from the virus or MS2 and temperature 4 and the bacterium. The genome size, this is the number of nucleotide base pairs in one copy of the total genetic information of the organisms, is indicated by the arrows near the upper numerical scale. The shape each of the renaturation curves is very simple and occurs with a single slope. However, the time over which renaturations occurs, which is very different and depends on the concentration of complementary fragments, which in turn depends on the size of the genome. The larger the genomes, the lower the saturation of complementary fragments in solution, and the greater the time required for renaturation to be complete. The second figure is the unidealized plot showing the kinetics of renaturation of eukaryotic DNA. When a single-strand DNA is allowed to renal, three classes of fragments can usually be distinguished by their frequency of repetition within the genome. A highly repeated DNA fraction, a moderately repeated DNA fraction, and non-repeated or the single copy DNA fraction. Take note that this is an idealized plot. The three classes of sequences are not a clearly separation in actual renaturation curve. A sequence arranged in this end-to-end manner is said to be present in tandem. Highly repeated sequences fall in several overlapping categories including satellite and DNA, many satellite DNAs, microsatellite DNAs. Satellite DNA satellite is consists short sequences or about 5 to few hundred base pairs in length that form very large clusters, each containing up to several million base pairs of DNA. In many species, the base composition of DNA segments is insufficiently different from the bulk of DNA that fragments containing the sequence can be separated into a distinct satellite that band during density gradient configurations. A species may have more than one satellite DNA sequence. The Drosophila virilis, for example, has three different satellites similar in sequence indicating a common genetic origin. The localization of satellite DNA within the centromeres of chromosomes is described in further that despite decades of research, the precise function of satellite DNA remains a mystery. Next is the mini satellite DNAs. It is the range from the about 12 to 100 base pairs in length and are found in the cluster containing as many as 3,000 repeats. Thus, many satellite sequences occupy considerable shorter stretches of the genome that those satellite is sequences. Many satellites tend to be unstable and the number of copies of a particular sequence often increases or decreases from one generation to the next. As a result, the length of a particular many-satellite locus is highly variable in the population, even among the members of the same family, because they are no variable or polymorphic, and the length many satellite sequences are used to identify the individual and criminal sequences are used to identify individuals in criminal or paternity cases through the technique of DNA fingerprinting. And last one is the microsatellite DNAs. This is the shortest sequences up to 1 to base pairs long and typically present in small clusters of about 10 to 40 base pairs in length. Microsatellites are excreted quite eventually through the DNA. More than 100,000 different microsatellite loci are present in the human genome. This figure is the DNA fingerprinting, and this technique, which is used widely to determine the DNA of the individual form of a simple DNA, and the DNA is digested by the treatment with specific nucleases. In the renailing experiments described, complementary DNA strands were allowed to be bind one of the another while colliding randomly in solution. An alternate experimental protocol called in situ hybridization was developed by Marie Laure Pardue and Joseph Gall of Yale University and was used initially to determine the location of the satellite. DNA, the term in situ means the in place, and which refers to the fact that the DNA of the chromosomes is kept in place while it is allowed to react with a particular preparations of labeled DNA in early in situ hybridization studies. The DNA to be detected was radioactive labeled and was localized by autoradiography. The resolution of the technique was been increased using probe DNAs or RNAs that are labeled with fluorescence dyes and then localized with the fluorescence microscope, and this latter technique is called fluorescence in situ hybridizations or FISH. and has become refined that it can be used to map the relative locations of different sequences along single DNA fibers. This figure is the fluorescence in situ hybridization and the localization of satellite DNA. A is the steps that are taken in carrying out fluorescence in situ hybridization, and this technique, the probe DNA is labeled without using radioactive isotopes. Instead, certain of the nucleotides in DNA are linked covalent to a small or organic molecule, usually butane. Following hybridizations, the location of the bound butane-labeled DNA can be visualized by treating the preparation with fluorescently labeled avidin, a protein that binds to biotin with a very high affinity. Chromosomes in this preparation usually appear red because they have been counter-made with a propidion iodide. The figure B is localization of a satellite DNA at the centromere of human chromosomes. The location of the bound biotin-labeled satellite DNA is revealed by the yellow fluorescent, which stands out against a background of red counter-made chromosomes. Fluorescence appears only at the site where each chromosome is constricted, which marks the location of the centromere. Like Fragile X syndrome, Friedreich's ataxia, Huntington disease, and more, is indicated by the location of each pyramid. The number of repeats are responsible for the normal red, carrier orange, and disease yellow. Conditions for each disease causing gene is indicated. Genes responsible for type 1 diseases such as Huntington do to exit the intermediate carrier state in which an individual possesses an stable allele but it is not affected. This figure is trinucleotide repeated sequences and the human disease. The top shows a generalized gene that is transcribed into an mRNA with several distinct portions or purple boxes, including a 5 non-coding portion called the 5UTR or a 5 untranslated region, a coding exon that carries information for amino acid sequence of the polypeptide, and A3 is non-coding portions. The introns in the DNA are not represented in the mature of RMNA. The general location of the nucleotide represents for each of 4 different diseases. Moderately repeated DNA sequences as the moderately repeated fraction of the genomes of plants and animals can vary from about 20 to more than 80% of total DNA depending on the organisms, which is the repeated DNA sequences with coding functions and repeated DNAs that lack coding functions. When we say non-repeated or single copy DNA sequences and initially predicted by the Mendel classical studies on the inheritance patterns of visible traits lead genetic to conclude that each gene was present in one copy per single or haploid set of chromosomes. Next, we discuss the stability of the gene, or the whole genome duplication polyploidizations. As you can see, this figure has twice the number of chromosomes as its cousins ectopicalis, or polyploidization is an event in which offspring are produced that have twice the number of chromosomes in each cells are their diploid parents. The offspring have 4 homologies of each chromosomes rather than 2. In 1971, Sosomo Ono of the City of Home Cancer Center in Los Angeles published a book entitled Evolution of Gene Duplication, in which they proposed that the evolution of vertebrates from much simpler invertebrate ancestor was made possible by 2 separate rounds of whole genome duplication during an early evolutionary period. Ono suggested that thousands of extra genes that would be generated by genome duplication that could be molded over time into new genes that were required to encode more complex vertebrate body. Over the past 3.5 decades, on a proposal has been hotly debated as genetics have tried to find evidence that supports or refutes the notions. As you can see in this figure is HAX genes, which are genes that play a key role in development of animals' basic body plan. Invertebrates that have examined have a single cluster of HAX genes. Our areas vertebrates have four such cluster, finding the supports ulnos hypothesis to runs and ancestral genome duplications. In a duplication and modification of DNA sequences have uh, duplications. Gene duplications refer to the duplication of a small portion of a single chromosome happens when the surprisingly high frequently and its occurrence is readily documented by genome analysis. And an equal crossing over is the duplication of the gene that probably occur by several different mechanisms, but it most often thought to be produced by a process. As you can see, and this figure is an equal crossing over between the duplicated genes provide a mechanism for generating gene changes in gene number. A is the initial stage shown has two related genes and deployed between individual gene 1 on the homolog can align with gene 2 on the other homologous during meiosis if crossing occurs. During this misalignment, half the gametes will be missing gene 2 and half will have an extra gene 2. And B figure is an unequal crossing over continues to occur during meiotic divisions in sequence generations. Randomly repeated array of DNA sequences will gradually evolve. Second figure is a pathway for evolution of globin genes. Exons are shown in red, introns in yellow. The evolutionary steps depicted in the diagram are discussed in the text. The arrangement of A, S, D, B globin genes on human chromosomes 16 and to 12 are the products of several millions years of evolutions. Hemoglobin molecules consist of two pairs of polypeptide chains. One pair is always a member of the A hemoglobin subfamily and the other pair is always a member of the B hemoglobin subfamily. A specific combination of ASDB hemoglobins are found of the different stages of development. The ASDB hemoglobin changes that are observed in embryonic, fetal, and adult hemoglobin are indicated. As you can see in this figure is the jumping genes and the dynamic nature of the genome. This is the visible manifestation of transpositions in maize. In 1940s, McClintock found that certain mutations were very unstable, appearing that disappearing from one of generation to the next or even during the lifetime of individual plant. That kernels of corn are typically uniform in color. The spots of this kernel result from a mutation in a gene that codes for an enzyme involved in pigment production. Mutations of this type can vary unstable, arising or disappearing during the period in which a single kernel develops. These unstable mutations appear and disappear as the result of movement of transposable elements during the period of development. In 1960s, several laboratories discovered that certain DNA sequences in bacterial move on the rare occasion from one place in genome together. Transposons were subsequently found to encode a protein or transposase the single-handed catalyze that excisions of a transposition from a donor DNA site and its subsequently insertion at a target DNA in transposition of bacterial transposition by the cut and paste mechanism. That figure is a sequence organization of a transposon. At the ends of the mobile transposable element contain a sequence or the read that is inversely repeated. Integration of the transposition into the host DNA creates a duplication that is seen as a short, direct, repeat or grid on the both sides or the element. The nucleotide sequence depicted here are those of TNR3, a bacterial transposon that codes of three proteins including the transposase that allows it is to integrated at the new sites in the DNA. In this figure is the schematic pathways in the movement of transposable elements. A figure is the DNA transpositions move off by a cut and paste pathways whose mechanism is depicted. Approximately 3% of the human genome consists of DNA transposition, none of which are capable of transposition. And B figure is the retrotransposons moved by a copy and paste pathways. The step involved in the retrotransposition takes place both in the nucleus, that cytoplasm, and required numerous proteins, including those of the host. More than 40% of the human genome consists retrotranspositions, only a few of which are thought to be capable of the transposition. More than one of mechanism retrotransposition is known. Several possible ways that transposable elements may have been involved in an adaptive evolution. First is the transposable elements can on occasion carry adjacent parts of the host genome with them as they move from one site to another. Second one is the DNA sequences that were originally derived from the transposable elements are found as part of the eukaryotic genes. And the third one is some cases transposable elements appear to the some given rise of genes themselves. One point is clear is the transposition has had a profound impact on the genetic composition of organisms. This point is clearly illustrated by a transposable element of the Zoophila melanogaster called the P elements. Okay, for the last subtopic is the sequencing genomes, the genetic basis on human being. The Human Genome Project will be effective completed in 2004. Number one, each site will, this is the fact that each site will have been sequenced on average about 10 times to ensure a very high degree of accuracy. Second fact is the sequences will contain a minimal numbers of gaps. And the darker matter is consistently larger along stretches high repeated DNA, especially those in around and centromeres of each chromosomes. In comparative genomics, if it is conserved, it must be important. Number one is the majority of the genome consists of DNA that resides between the genes and thus represents intergenic DNA. The second one, each of 30,000 protein coding genes consist largely of non-coding proteins or intronic DNA. This figure is the small segments of DNA are highly conserved between humans and related species. The top line represents the nucleotide sequence of the segments of human DNA. Each line below it represents the nucleotide sequence from one primate as indicated on the right. Those blocks on which each line represents segments whose nucleotide sequence varies from that of the corresponding human segments. The bottom line shows those parts of these DNA segments that are highly conserved in all of the species shown that is those parts whose sequence does not vary in any of the 8 species represented. Another important difference in the fact that a common human epithelial cancers like breast and colon cancer are only rarely seen in chimpanzees. To date, the greatest interest in this field has focused on a gene known as FOXP2. A comparison of FOXP2 proteins between humans and chimps shows two amino acids a difference that have appeared in a human lineage since the time of separation from one or less common ancestors. FOXP2 gene suffer from a severe speech and language disorder. Among other defects, persons with disorder are unable to perform the fine muscular movements of the lips and tongue that are required to engage in vocal communication. Calculations guess it from the chimp's versions became fixed in the human genome within the past 120,000 to 200,000 years, a time when modern humans are thought to have emerged. This finding suggests that changes in FOXP2 gene may have played an important role in human evolution. The impact on the medicine of the human genome sequences is discussed in the accompanying human perspective. In a human perspective, it is the application of genomic analysis to medicine. Genetic polymorphisms or sites in the genome that vary among different individuals. It refers to a genetic variant that occurs in at least 1% of a species populations. And a single nucleotide polymorphism is NPs. The common type of genetics variability in human occurs at sites in the genome where single nucleotide differences are found among different members of the populations. In this figure shows that the genome is divided into block or haplotypes. This top line shows hypothetical segment of DNA containing a number of SNPs. Each SNP is indicated by a filled circle. This particular segment consists of 5 haploids separatedly by short stretches of DNA that are highly variable. Each haploid occurs as a very small number of variants. Each haplotype shown there here exists as 3 to 6 different variants. Each haplotype variant is characterized by a specific or set of SNPs. Indicated by the colored circle, all of the SNPs of a particular haplotype variant are drawn in the same color of indicated that they are inherited as a group and found together the different members of the population. Each of persons represents at the bottom of the illustration as a particular combination of haplotypes on each and their 2 chromosomes. Some haplotype variants are found in many different ethnic groups, others have more restricted distributions. Next is the experimental pathways, the chemical nature of the genes. 2 years after Gregor Mendel presented the results of his work on inheritance in pea plants, Friedrich Mescher graduated from a Swiss medical school and traveled to Tübingen, Germany, to spend a year studying under Ernest Hope Seyler, one of the foremost chemists and possibly the first biochemist of the period. Messer was interested in the chemical contents of the nucleus to insulate pepsin. The residue from the treatment was composed primarily of isolated cell nuclei that settled to the bottom of the vessel. Messer then extracted the nuclei which dilute alkali. Messrs found the alkaline extract contained a substance that had proteins unlike any previously discovered. The molecule was very large, acidic, and rich in phosphorus. He called the material nuclein. His year, Messier returned home to Switzerland while Hoppy Zaylor, who was continuous about the findings, repeated the work with the results to confirm. Messier published the findings in 1871. In fact, that they were the first preparations of relatively pure DNA. The term nucleic Messier students who work out the methods of purifying propane-free DNA from various animal tissue and yeast. Botanist name is Zacharias Visible also stained a preparation of nuclein that had been extracted using a Messier procedure of digestion with pepsin is in an HCI medium. Further, when the pepsin HCl extracted cells were subsequently extracted with a dilute alkali. A procedure was known of the removed nuclein, the cell residue, which included the chromosome remnants, no longer contain stable material. This and other results pointed strongly to nuclein as the component of the chromosomes. In the remarkable Frashenten proposal of Otto Hetwedge. Some of the most important contributions in the pursuit were made by the Piobist Aaron Leviny, who immigrated from Russia to the United States in 1891 and eventually settled in positions at the Rockefeller Institute in New York. It was Leviny who finally solved one of the most resistant problem of DNA chemistry when he determined in 1929 that the sugar of the nucleotides was too deoxyribose to isolate the sugar 11 and ES. London placed the DNA into the sample from the animal's intestine as it passed through the stomach and intestine various enzymes of the animal's digestions that in digestive tract active on the DNA carrying the nucleotides into their components part, which could be then isolated. That analyzed Levin summarized the state of knowledge on nucleic acid in the monograph published in 1931. That who had been studying the behavior of the chromosomes during fertilization stated in 1884 that he said, "I believe that have at least made it highly probable that nuclein is the substance that is responsible not only for fertilization but also for the transmission of hereditary characteristic." Ironically, as more was learned about the properties of nuclein, that the lysine was considered as the candidate from the genetic material. That figure is these large glistening colonies on the right and virulent S-type pneumococci, whereas the small colonies on the left are non-virulent R-type pneumococci as discussed below. The cells in these particular S colonies are the result of transformation of R bacteria by DNA from heat-killed S. pneumococci. In 1923, Frederick Griffith, a medical officer at the British Ministry of Health, demonstrated the pneumococcus also grow as either S or R colonies, and furthermore, the two forms were interconvertibly ball, that is, an occasions an R bacterium could revent to an S form or vice versa. Griffith found, for an example, of the injected exceptionally large number of R bacteria into a mouse. The animal frequently developed pneumonia and produce bacteria that form colonies of the S form. And every every had initially been skeptical of the idea that as a substance released by a dead cell could alter the appearance of a living cell. But he was conceived when the Martin Dawson's, a young associated in his lab, confirmed the results. Dawson went on to show that transformation need to occur within a living animal host. Culture uh, that growth struck of the dead as bacteria when mixed in a bacterial culture with a small number of a non-virulent or R cells in the presence of anti-R serum was capable of converting the R cells into virulent S form. The transformed cells were always of the type 1, 2, or 3 characteristic of the dead S cells. The next major step was taken by J. Lionel Alloway, another member of Avrislav, who had able to solubilize the transforming agent. This was accomplished by rapidly freezing and throwing the killed donor cells, then heating the disrupted cells, centrifuging the suspensions and forcing the uh, supertans through the porcelain filter whose pures block the passage of bacteria. The soluble filtered extract possess the same transforming capacity as the original heat-killed cells. All evidence suggested that the active substance was the DNA. First is it exhibited a host of chemical properties that were characteristic of DNA. Second one is no other type of material could be detected in the preparations. The third one is the test of variations enzymes indicated that only those enzymes that digest the DNA and active the transforming principle. This figure shows the outline the experiment by Griffith of discovery of bacterial transformations of the mouse. This is McKinley McCarthy, one of the three authors described in Ancient in 1949 when he was asked to speak at Johns Hopkins University along with Leslie Gay, who had been testing the effects of the new drug Dramamine from the treatment of the seasickness. This figure shows the electron micrograph of bacterial cell infected by T4 or bacteriophage. A phage is seen attached by its tail fibers to the outer surface of the bacterial cell, while new phage heads are being assembled in host cell's cytoplasm. This figure is the Herschel chest experiment showing that bacterial cells infected with a phage containing P labeled DNA or red DNA molecules became radioactive labeled and produced labeled phage progeny. In contrast, bacterial cells infected with prophage containing S labeled protein or red phage coats did not become radioactive labeled and produced only non-labeled progeny. This is the synopsis of our report of Module 4 Genetic Information. I'll posted some videos that are related to the genetic informations of our report. Please visit the Google Classroom. Thank you for listening.